begins in the 19th century with Charles Darwin and Alfred Russel Wallace. In 1858, their ideas on natural selection were presented together to the Linnaean Society, and in 1859, Darwin expanded the argument in On the Origin of Species. Darwin's key insight was that small, heritable differences, filtered by competition and survival, could accumulate into major evolutionary change without any need for sudden creation of new forms. What Darwin still lacked was a reliable explanation of how inheritance itself worked. That missing piece came from Gregor Mendel. His 1866 work on pea plants showed that inheritance is particulate, not a smooth blending process. Traits are passed on in discrete units, now understood as genes, and those units can segregate and assort independently. Mendel's work was not immediately integrated into evolutionary theory, but once rediscovered around 1900, it solved one of Darwin's biggest problems: how variation can persist instead of being diluted away. During the first half of the 20th century, evolutionary biology was rebuilt as the modern synthesis. Population genetics showed mathematically how Mendelian inheritance and natural selection fit together. Fisher, Haldane, and Wright developed the theory of how gene frequencies change in populations, while Dobzhansky, Meyer, and Huxley helped connect that mathematics to real organisms, species formation, and the fossil record. The result was a unified framework in which evolution became change in allele frequencies through mutation, selection, drift, migration, and recombination. In that framework, mutation creates new genetic variants. Recombination reshuffles them. Natural selection increases the frequency of variants that improve reproductive success. Genetic drift changes frequencies by chance, and gene flow moves variants between populations. Speciation happens when populations become sufficiently isolated that their gene pools diverge and reproductive barriers build up. In other words, evolution is not a ladder or a plan. It is a population-level process in which many small changes are sorted over time by environment, chance, and biological constraints. From the 1960s onward, molecular biology changed the field again. Moto Kimura's neutral theory argued that a large fraction of evolutionary change at the molecular level is not driven by selection at all, but by random fixation of selectively neutral or nearly neutral mutations. That did not replace Darwinian evolution. It refined it. The lesson was that selection is crucial for many traits, but not every change in DNA is adaptive. Some changes are neutral, and in finite populations, those changes can spread or disappear through drift. As sequencing became cheaper and faster, evolutionary science moved from single genes to whole genomes. High-throughput DNA sequencing now lets researchers reconstruct phylogenetic trees from genome-wide data, and target capture methods have expanded the usable evidence base by turning even museum and herbarium specimens into sequenceable material. This has transformed systematics, comparative biology, and the reconstruction of the tree of life, because scientists can now compare far more DNA across far more species than ever before. The same genomic revolution has changed the study of human evolution. By comparing modern genomes with those of archaic hominins, great apes, and other primates, researchers can identify human-specific genetic changes and connect them to cell types, developmental stages, and organoid models. Ancient DNA has taken this further by allowing scientists to observe genetic change through time rather than infer it indirectly from the present. That means adaptation, population movement, and natural selection can now be studied with far greater temporal resolution. Another major shift has come from evolutionary developmental biology, often called Evo-Devo. This field studies how developmental programs shape the kinds of variation that evolution can act on. It asks not only which mutations matter, but also why some body plans and traits are easier to evolve than others because of developmental constraints, gene regulation, and timing. In the current literature, Evo-Devo is often treated as a complement to the modern synthesis rather than a replacement for it. Experimental evolution has also become central. In long-term laboratory experiments, especially with microbes and yeast, scientists can watch evolution happen in real time across many generations. These studies reveal mutation rates, repeated adaptive solutions, trade-offs, changing fitness landscapes, and the ways populations respond to shifting environments. This is one of the strongest proofs that evolutionary theory is not just historical interpretation. It is a predictive framework that can be tested experimentally.